recording. Now I am recording. So the week 5 lectures on haemoglobinopathies, lecture 1 thalassemias, lecture 2 will be on haemoglobin variants. So as usual, there are a number of learning objectives that will be covered by the lectures, the tutorial tomorrow, and also by the pracs on Thursday. As usual, we use the same sort of process that we've been using when we are looking at the looking at our other health conditions that we've been looking at during the semester, and this time we are applying it to people who have a haemoglobinopathy. So you need to know the general characteristics of the condition from a clinical point of view and a laboratory point of view, and to understand why you're seeing what you're seeing in the laboratory and why the people have the clinical signs and symptoms that they do. We need to understand what causes the condition, so the etiology of the condition, the pathogenesis, and also the pathophysiology. So why we're seeing those abnormal changes that we're seeing. You only really need to know about any prognosis or treatment that actually has a laboratory component to it. So if it's where you know, the laboratory is monitoring the treatment or monitoring the condition in some way. And coming back to our classification of our anemias, and of course they're classified on the MCV, on the cell size in the first instance. And in the first few weeks we were looking at normocytic anemias. So these are anemias that had a normal MCV within the reference interval. Last week we looked at macrocytic anemias, both megaloblastic and non-megaloblastic, and this week is our first foray into looking at microcytic anemias, which the thalassemias are, and you can see them in the list on that slide there down here. And also don't forget that our retic count is also our very useful indicator for helping with our classification as to whether the retic count is increased or decreased. Now, it's not of that much significance when we're talking about the haemoglobinopathies, although we will see in the second lecture today that we can have an increased retic count when we start talking about haemoglobin variants. So again, we have a definition for our microcytic anemia, and of course it is around the MCV, but as you can see up the top there, it is a quantitative decrease in hemoglobin synthesis which results in microcytic erythrocytes. So basically what's happening here is that for some reason synthesis of hemoglobin has slowed down, has decreased, and so there's not enough hemoglobin to properly fill the erythrocytes. And usually the way the bone marrow works is that when there is a reduction in a particular component that is needed for erythropoiesis in particular, then the cells tend to get smaller first and then they become hypochromic. And so these microcytic erythrocytes usually, as we will see going on, certainly for iron deficiency anemia, but in most thalassemias we see that they are hypochromic as well, but not always. But they are all microcytic. So with an MCV of less than 80 femtoliters basically, although I know that our reference interval actually goes down to 78. Unlike our macrocytic anemias that we talked about last week, or with our megaloblastic anemias, DNA synthesis and cellular division are not impaired. So that is still everything, the mechanics in that way is still fine. It's just that something is preventing hemoglobin from being produced. And so we can have impaired globin synthesis, which is what we see with thalassemia, or we can have disordered globin structure. So that our hemoglobinopathies are all to do with something happening with the globin part of the hemoglobin molecule. When we have a look at disorders of iron metabolism next week, we will see that iron is all about heme synthesis. So this is where it's very important for you to recall the structure of the hemoglobin molecule, and so that you recognise that we've got the heme discs where the iron is held, and also the 4 globin chains. So the 2 pairs of globin, protein globin chains that actually all contain a heme disc. So when we're talking about the hemoglobinopathies, we're talking about abnormalities to the globin chains. The difference between thalassemias and structural hemoglobin variants is quantitative versus qualitative. When we're looking at the thalassemias, all thalassemias have some sort of a quantitative defect in globin chain production. So we've got less globin chain production and therefore less hemoglobin in the erythrocytes, therefore smaller and paler cells. So this is why with the thalassemias we have microcytic anemias. Whereas when we look at the structured hemoglobin variants, the normal amount of hemoglobin is being produced, but it's abnormal hemoglobin. So we have altered globin chain structure. And so that's why the majority of structural hemoglobin variants are normocytic. So usually normocytic, normochromic. And so we find that when we have a look on our classification system, as I said before, the hemoglobin variants are normocytic, normochromic anemias, and they are also classed as a hemolytic anemia. So that abnormality to the hemoglobin structure and the abnormality to the hemoglobin function mean that these cells are more fragile and therefore they don't live their usual 120 days. And usually the structural variation does not affect the rate of synthesis of the affected globin chains the way that a quantitative effect can affect the rate of synthesis. So we usually have normal rate of synthesis of the chains. But we have altered chain structure, therefore we have altered chain function or altered hemoglobin function. So you're getting used to having a look at this slide by now. We saw it, I don't know when we saw it last. I think it was when we were looking at malaria. But you can see here that the thalassemias and the abnormal hemoglobin variants have a very, very similar distribution globally to malaria. And again, this is suggesting that evolutionary pressures have allowed the genetic defects to continue through the generations because they confer a benefit onto people being able to be more resistant to malaria. So thalassemia in the blue, you can see going right along from the Mediterranean through through the Middle East and then along through India and Southeast Asia. Whereas the hemoglobin variants tend to be a little bit more patchy in their distribution. Hemoglobin S or sickle cell anemia, the pink, is most widely distributed, and then hemoglobin E, very common in Southeast Asia. Hemoglobin D has just that little pocket in West India there, and hemoglobin C is in West Africa. They're roughly about the place where Plasmodium ovale replaced Plasmodium vivax, so that's quite interesting, isn't it? Mackenzie has a lot of really, really useful tables and summaries around the thalassemia and the structural variants. So in chapter 14, it's a very good chapter in that respect, because there is a lot of information around the hemoglobinopathies, and I really like the way they have structured these tables. So you can see here that With the structural variants, we usually get a decrease in our red cell count, whereas with the thalassemias, we actually get an increase in our red cell count. And that's quite unusual, because I'm sure you've realised by now that over the last 4 weeks, anytime we have a low hemoglobin, we've usually had a low red cell count. And this is not the case with many of the thalassemias, particularly the more subclinical types, or the less nasty types. They tend to have a red cell count that is either normal or increased, and certainly doesn't reflect the level of the hemoglobin. But we'll talk more about that later. We can see target cells in almost everything that we look at, but there are some very specific red cell morphologies and inclusions that we see with the structural variants. And don't forget that basophilic stippling that we saw in that rather lovely film from last week with the person who had beta thalassemia. And there are ways in which we can actually detect the abnormal hemoglobin, and there we have whereabouts we usually find these conditions. And please notice, as I said before, that we tend to get an increased retic count with our structural hemoglobin variants, because they are normally have more of a hemolytic component to them than the thalassemias do. Yes, the cells are more fragile in thalassemia, but we don't tend to get that same response of increased reticulocytes with the thalassemias that we can get with our structural variants of hemoglobin. So when we're looking at the causes and the progressions of conditions with thalassemia, it was first described in 1925 in a family of 4 children who all exhibited symptoms of anemia, hepatosplenomegaly, so that's enlarged liver and enlarged spleen, and altered facial features. And so these altered facial features were, remember I've talked before about extramedullary hemipoiesis, particularly happening in children when the normal bone marrow, the red bone marrow of our axial skeleton, can't keep up with hemoglobin production. And so we start producing hemipoiesis in other sites in the body. And one of the more common of these sites is in the flat bones, because the flat bones are not needed for support. And so we tend to get abnormal facial features because it's the flat bones of the skull that are, if you like, recruited, and we get additional hemipoiesis happening there. So I've just noticed that I've got a couple of rather weird looking signs on this slide. So that first sign that you see there, unbalanced synthesis of, that should be an alpha there, of alpha or beta chains, or fetal gamma chains. So you might like to make a note of that on your notes. That should be alpha, that should be beta. So the thalassemias are the most common recessive diseases worldwide, and they're quite a heterogeneous group of haematological disorders. And so we can get quite a wide variety of clinical presentations. And thank you very much, whoever did that for me. And a wide variety of what we see in the laboratory as well. So it's quite a common genetic disorder, particularly the heterozygous forms. And so both thalassemia and hemoglobin variants are autosomal recessive conditions. So you can have homozygous, you can have heterozygous, you can also have compound heterozygous, where the lucky individual doesn't just inherit one abnormal form of hemoglobin from one parent, but inherits a different abnormal form of hemoglobin from the other parent. So they're compound heterozygotes. But the 2 main variants of classical thalassemia are alpha thalassemia and also beta thalassemia. Alpha thalassemia is quite common in Asia, the Middle East and the Mediterranean region, whereas beta thalassemia tends to be more localized to the Mediterranean population. And so what we tend to get is this decreased in total hemoglobin because we've got reduced or prevented synthesis or one or more of the constituent globin chains. So we get a decrease in total hemoglobin, but we also get unbalanced synthesis of the chains that make that hemoglobin. So remember that in our hemoglobins, we have got, as I said before, two pairs of chains. So in hemoglobin A, we have alpha chains and beta chains. In hemoglobin A2, we have alpha chains and delta chains, and hemoglobin F, or fetal hemoglobin, we have alpha chains and gamma chains. And so when we get reduced synthesis of one of those chains, it means that the other half of the pair doesn't have anything to pair up to. And so we get precipitation of these chains, or we can get different sorts of hemoglobins being produced from these chains transforming into tetramers. Because reduced or prevented synthesis, what can happen is that one of the genes might be defective, but the other one's okay, and that gene still keeps on producing hemoglobin, although it may not be able to produce quite as much. And those unpaired globin chains, of course, are able to damage the red cells, and so we have fewer cells actually leaving the bone marrow, so we do get an aspect of ineffective erythropoiesis, and so some forms of thalassemia can actually be quite severe. So there are two genes situated on chromosome 16 that encode for alpha chain synthesis, and one that sits on chromosome 11. So it's quite interesting that we've got two for alpha. It's like we've got an extra one for just in case. And on chromosome 11, you can see we've got one gene for each of the other hemoglobins. And remembering that we inherit one chromosome parent, so we end up with 4 genes for making alpha chains and 2 genes for making the other chains that are there. And so you can see that it's a combination usually, except for in the very early stage embryo up to about 9 or 10 weeks, where we get these hemoglobin Portland and hemoglobin Gao are being produced. But otherwise, it is alpha chains with something else. So it means that if we get alpha thalassemia, it's really can be quite horrendous because we need alpha chains for each of our adult hemoglobins. Whereas if we get beta thalassemia, we've got 2 fallback hemoglobins, if you like, and we usually get reduced amounts of hemoglobin A being produced and increased amounts of hemoglobin F and hemoglobin A2. But alpha thalassemia would appear to be the far more serious condition, but remember that we actually have 4 alpha genes that can encode for hemoglobin. So, you know, we've got, I guess, 4 chances of being able to make some hemoglobin. Oh dear, I am sorry. So you should have notes that you have downloaded from Echo 360. So in this first line here, we're looking at alpha thalassemia genotype. So every time where you see this beautiful symbol, that should be an alpha. And so when we see alpha thalassemia, it is a decrease in gene types. And so it's a decrease in the chains, in the globin chains, and it's a deletion of a gene, as you can see here. And at the top here is the most severe form where we have all 4 alpha chains have been deleted. Thank you very much, person. And then down the bottom here, you can see that we've got, this is normal, where we've got all of our 4 alpha chains here. So these two here, these alpha thalassemia minor, they tend to be subclinical, and this one here, this alpha thal trait in blue, tends to be subclinical as well. So the 2 genes that are there or the 3 genes that are there, are able to produce enough alpha chains that we're able to live quite normally with very few clinical signs or symptoms. And so these people here, the alpha thal traits and the alpha thalassemia carrier here, they're usually picked up either in population screening or serendipitously. So again, it's our hemoglobin or FBE check for preoperative check, or a blood donor check, or a medical condition check, medical insurance check. It's these ones here, alpha thalassemia intermedia, where we only have one alpha chain being produced, and of course alpha thalassemia major, where we have no alpha chains being produced, that we get the serious signs and symptoms. In fact, alpha thalassemia major usually results in the death of the fetus in utero, or very shortly after birth. And so you can see here that we have hemoglobin Barts here, and so we have no alpha chains being produced, but in the fetus or in the embryo, we get an excess, fetus and embryo, we get excessive gamma chains, and then once the child is born, as in alpha thalassemia intermedia, we can get beta globin chains being formed, and that forms something called hemoglobin H. So hemoglobin H is when we have beta tetramers, so we have 4 beta chains that have combined together. Hemoglobin Barts is where we have 4 gamma chains that have combined together. The one that's in blue here, alpha thalassemia minor, that is the most common form of alpha thalassemia minor seen amongst the Chinese, Thai, and Middle Eastern ethnicities. And so, and that's fine. There's enough for the person to be quite happy. But as you can see, if you have 2 people who have this particular genotype with the alpha alpha and delete delete, if they have a child, then the chances of that child having alpha thalassemia major are 50%. And so it is quite important in these areas, if there is a thalassemia history within the family, for genetic counselling and genetic screening to be done if people are thinking of having children. So just these next couple of slides are just looking at the pathophysiology and clinical consequences of each of the alpha thalassemia conditions. So we've got alpha thalassemia major, and as I said, usually the fetus does not survive, and because it only has gamma chains that can form together to form the tetramers. And the tetramers are quite stable, but they have an increased oxygen affinity, and so we don't get proper oxygen supply to the tissues, and the fetus usually dies in utero, and it's another cause of what we call hydrops fetalis. So this is where the fetus actually dies from heart failure, and this heart failure leads to edema. And so the fetus looks like it's full of water, basically. That's what the term means. At the other end of the spectrum, we've got alpha thalassemia minor. And as I said, this second one here is the more common phenotype that we see, or genotype that we see, sorry. And we have reduced alpha globin chain synthesis, but we usually, in the fetus, you might get some hemoglobin Barts being formed, but when that gamma beta switch happens at about 6 months of age, then we have the hemoglobin H isn't usually present, because the interesting thing about alpha and beta chain production is that the alpha chain production tends to limit the rate or affect the rate of the beta chain production. So in these situations of alpha thalassemia minor, we don't tend to get the excessive beta chain tetramers being formed, and so we don't get hemoglobin H in those situations. And then we've got alpha thalassemia intermedia, which is halfway between the two, funnily enough. And so this is the one where we see quite a few clinical signs and symptoms, and also the laboratory sign. So this is probably, oh no, I was thinking this is what we had last week, but no, it's a beta thalassemia we had last week. So this is what's known as hemoglobin H disease because it's picked up very early on in newborns, and because again they're not very well and they usually have a hemolytic anemia. And so hemoglobin Bart's is the most common hemoglobin that is found there, in the highest concentration, and of course it's the one that causes the tetramers that we see that precipitate out, and we can't see it as hemoglobin Bart, sorry, we see it here. This hemoglobin H is once the gamma beta switch has happened, and then we do get the excess beta chains in this situation because there is so little alpha globin being produced. And so that's why it's called hemoglobin H disease, because we get these hemoglobin H beta tetramers, and we can actually see these on when we use a supravital stain. You can't see them normally in a normal blood film, but you can see them using a supravital stain. So we have mild to moderate compensated hemolytic anemia with very few clinical signs or symptoms, but once they get into an oxidative crisis, that's when we come into problems, because the beta tetramers are not able to properly oxidate, properly perfuse the tissues. So again, this is a situation of hemolytic crisis, just like we get with our G6PD. So on to thalassemia, beta thalassemia. Unlike alpha thalassemia, beta thalassemia is due to point mutations. Alpha thalassemia is gene deletions, whereas beta thalassemia is point mutations. And this means that we have a gazillion different sorts of beta thalassemia. And as you can see there, it affects any step in the pathway of expression. This is also why with beta thalassemia you can get all these sort of compound heterozygotes. Thank you. Most commonly we have partial or complete absence of beta gene expression, but we can also have a decreased rate of beta globin. They can be being produced, but they're at a decreased rate. When we have beta thalassemia, because we have fewer beta chains being produced, this means that we have unpaired alpha globin chains. So just as unpaired beta globin chains produce hemoglobin H, unpaired alpha globin chains can also precipitate, but they form Heinz bodies. So they are another way in which we can have Heinz body formation. So we have Heinz body formation in G6PD deficiency. We have Heinz body formation in beta thalassemia. Again, we have a wide range of clinical expression depending on the rate of synthesis of the beta globin chains. And again, you can see when we've got beta thalassemia here, we inherit one beta globin chain gene from each parent, so we only have the 2 genes there. But you can see here that only hemoglobin A has beta chains as the pair to the alphas, and so what we tend to find is that we have increased concentrations of hemoglobin A2 and increased concentrations of hemoglobin F. And so we are able to detect the reduced amount of hemoglobin A and the increase in concentrations in these two. Now on this page, of course, all of those beautiful little images are meant to be betas there. It's interesting that a bee came up there. And so again, you can see that we have beta thalassemia major going down to a beta thalassemia silent carrier. And of course, normal down the bottom is when we have inherited 2 normal beta genes, 1 from each parent. So the beta naught that you see here, that means it's a gene mutation in which there are no beta globin chains being produced. So of course, if you have had the luck to inherit 2 beta naught genes, then you're going to have a fairly major condition. The beta plus that you see here is a 5 to 30% decrease in the production of the beta globin chains, and the beta silent is there's only a mild decrease, so less than 5% of a decrease. So that's why, again, with these conditions here, so thalassemia intermedia and thalassemia minor, some of these are quite asymptomatic. So as I said before, because we've got a decrease in the beta globin chains, we have an increase of synthesis in the delta and gamma chains. So we have an increase in hemoglobin A2 and hemoglobin F. However, we still do have ineffective erythropoiesis because of these unpaired alpha globin chains. And so they precipitate and cause damage to the red cell membrane, and so many cells do not get to leave the bone marrow, or those that do can be more fragile. Again, it depends on how much the beta globin synthesis has been affected. But again, in extreme situations, such as in the children that in which hemoglobinopathy or thalassemia was first diagnosed, they had quite serious extramedullary erythropoiesis, particularly in the bones of their face. Depending on the type of thalassemia that you have, and this tends to be more so with beta thalassemia than alpha thalassemia, you can have transfusion independent forms and transfusion dependent forms. So that's when people have to have regular transfusions to be able to survive. The issue here with the transfusion dependent forms is iron overload. And so iron overload happens because you are bypassing the gut. And so you may recall from your normal iron metabolism that you learned about last year that it's all about the absorption of iron that's the rate limiting process. And if you bypass that, the gut, so the enterocytes in the gut, then you are feeding iron straight into the blood, and all of the cells in the body will say thank you very much, and they will take up that iron. So iron overload is an issue with these people. With alpha thalassemia, we don't tend to get as many who need to look to transfusion. I think it's with alpha thalassemia, you can either live with it or you can't, basically, whereas beta thalassemia is a lot more variable. And these are the sorts of deformities I was talking about. So here are the flat bones of the skull, and so they have been reverted back to hemipoiesis, and so the insides of the bones, so that the spaces have been expanded, and this is what is causing these abnormalities in the skull bones. And you also find that you get softening of the bones, particularly of the femur, and you can see also here that the cortex of the bone is is quite thin, because the red marrow in the middle here has expanded, because it's trying as hard as it can to keep up with the demand. Again, this is another table from Mackenzie, and so it again quite nicely summarises in general the clinical presentation, the pathophysiology, and what we see in the laboratory. So has anybody got any questions up till now that you'd either like to ask verbally or that you'd like to write? stunned silence. So, oh, here we go. Thank you, Kiani. So Kiani doesn't have any questions. So if we go to the next slide here. So this is, as usual, I leave this slide bare for you to fill in because that's really important for you to think about the populations that are most at risk when we are looking at who is most likely to have thalassemia. Now, I've even got a little polling function in Webroom, you'll be very pleased to know. So what I thought I would do is put up a couple of learning checkpoints. So if you bear with me just while I type them in. Okay, so thalassemias are classified as conditions of decreased globin production, decreased heme production, or variation in amino acid composition. Okay, so the answer to that is the first one, of course, which is a decrease in globin production. So the point mutations that we see in beta thalassemia, they still do result in a decrease of globin production. So that's why they are still classified in those conditions. Okay, so then the next one. So alpha and beta thalassemia are differentiated by the ethnicity of the population affected, the age of the population affected, the type of globin chains affected, or the abnormal hemoglobins produced. Okay, so you seem to be more willing to take part in this poll compared to when we do it through Echo 360. So yes, it is the type of globin chains affected. That is the difference between them. In alpha thalassemia, we have the alpha globin chains are affected, and beta thalassemia, we have the beta globin chains affected. And finally, for my last poll here, okay, alpha thalassemia is a far more serious condition than beta thalassemia, not blank square. because all forms of normal hemoglobin contain alpha chains, because alpha chains precipitate to form hemoglobin H, causing hemolysis, because beta chains precipitate to form hemoglobin Barts, causing hemolysis, because mutations in beta chain genes are more stable than those in alpha chain genes. So the majority of you are correct. All forms of normal hemoglobin contain alpha chains. So if you remember, alpha thalassemia major is where we lose all of the alpha genes. So we have a deletion of all 4 genes. And because we don't have any alpha chains, that means that no hemoglobin can be produced. And so it's a fetus that has inherited 4 defective genes in this way is usually dies, and that's where we get hydrops vitalis. Hemoglobin H, they are beta chains that precipitate to form hemoglobin H. Okay, well done. So we'll go on and have a look at the laboratory results that we see in thalassemia. And again, this is very much a summary, and as I've said all the way along, really it depends on the severity of the condition as to what you're going to see in the laboratory. So if the condition is a clinical condition, then you will see changes. And the majority, or they will all be microcytic anemias that we see, and some of them will be hypochromic. It really does depend on whether or not you've got a thalassemia trait or whether or not you've got one of the more severe forms, so the intermediate or the minor. The really interesting, or the really important thing to note here is, as I pointed out before, with the thalassemias, even though the hemoglobin can be reduced, the hemoglobin concentration can be reduced, the red cell count may not be, depending on the form of thalassemia that you have. And so if you come across a case study where you have a low hemoglobin with a normal or increased red cell count, the first thing that should jump into your brain is, I wonder if this person has thalassemia. Particularly one of the more mild forms, and particularly if you've got a low MCV, because thalassemias are all microcytic. Not all of them are hypochromic, but they're all microcytic. The ones that are hypochromic, of course, we have to differentiate from iron deficiency, as I said before. You will see red cell inclusions if you have got the more clinical forms of alpha thalassemia. You will have the unpaired beta chains precipitating and forming hemoglobin H. And if you've got one of the more clinical forms of beta thalassemia, then you will get the basophilic stippling. So the basophilic stippling is not alpha globin chain precipitation. It's more the degradation of the cytoplasmic RNA that's causing that stippling. Basophilic stippling, everybody saw last week, I think, in our Magrum-Wilgenza film. Hemoglobin H you only see in the supravital stains. So that's the same sorts of stains that we use when we are staining for reticulocytes. So alpha thalassemia intermedia is the one that we call hemoglobin H, and this is the one where we get the unpaired beta chains. So you can see here, so this is the one where we've got only one working alpha chain, if you like, the deletion of the 3 other chains, and so you can still see that the hemoglobin isn't too bad. So hemoglobin it's really only a mild to moderate anemia, and the red cell count, notice, is normal or increased. So hemoglobin is down, but the red cell count is normal or increased. The MCV is less than 75 usually, so it's usually quite microcytic, and the MCH is down as well. And so even though the cells are small, there's usually not enough hemoglobin in them for the size of the cell, and that's why our MCHC can be low. You can see that poikilocytosis and anisocytosis are common, and of course target cells are there as well. So the further tests that we use for alpha thalassemia intermedia, so we can stain for hemoglobin H. That's a really good thing to do, because it's quite difficult with alpha thalassemia, because we have alpha chains in all of our hemoglobins. When you do a hemoglobin electrophoresis, it's often quite difficult to see any change, because usually with an alpha thalassemia, you'll find that all of the hemoglobin, the adult hemoglobins are lower. So hemoglobin F, hemoglobin A2, and hemoglobin A. But interestingly, there does seem to be a little bit of an increase in hemoglobin A2. Not much, but there is a little bit of an increase. And also hemoglobin H, so the beta chain tetramers, they do actually show up on hemoglobin electrophoresis. And of course, then we would go to genetic testing. And this is the sort of thing that we'd see in alpha thalassemia intermedia. So you can see that these cells are quite hypochromic. There's quite a variation in their size, from being sort of vaguely normal to being quite little. And you can also see some variety in the cell shapes as well. And this slide here is just showing you the supravital stain for the hemoglobin H. It's very pale, but hopefully you can see that there is dots all over some of these cells, and it looks very similar to the basophilic stippling, doesn't it? But you have to do supravital staining to see hemoglobin H. So now going on to beta thalassemia. Again, we've got the 4 categories that we see. So we've got the silent carrier, beta thalassemia minor, and then beta thalassemia intermedia, and beta thalassemia major. So once again, you can see that we've got this progression of what we can see in the laboratory, from no hematological abnormalities at all. That's for the silent carrier. That's why it's called that, because you don't know that it's there unless you would be doing population screening for abnormalities. And then we've got beta thalassemia minor. So this is the beta plus, where we have 5 to 30% reduction in hemoglobin concentration. And then this one is, let me see, I'll just have a look for you in my notes, what that one's meant to be. So that's meant to be beta plus and beta, so that's the normal beta chain, and beta naught, where there is less than 5% being produced, and the normal beta there. So you can see here we get mild hemolytic anemia, so it's quite mild really, and again normal to slightly increased red cell count. So that's what's so important, to check your FBC results when you are looking at your case studies, because there are certain little characteristics that will tell you that you could have a particular condition. So beta thalassemia minor is where we get the basophilic stippling, and we basophilic stippling in our Magwin-Genza slides, our normal slides. So it was thalassemia intermedia that we had last week. So we had microcytic hyperchromic anemia with marked anisocytosis and marked poikilocytosis. So we had all of these things here, plus the nucleated red cells, didn't we? And then thalassemia major, it's even worse. and you get basophilic stippling, Howe Jolly bodies, and also Pappenheimer bodies, which I think some people saw. And so this is thalassemia minor here, and I've just circled these two cells because they've got a tiny bit of basophilic stippling in the hemoglobin that you see in them, and then that's thalassemia major with lots and lots of red cells there. So this flowchart is from a paper that I've got the citation to on the reference slide, and it's a lovely little flowchart of how you go about identifying thalassemia. So as usual, we start with our clinical history plus our standard screening tests, so our full count, full blood examination. So it's all to do with the MCV and the MCH. And then if we have a microcytic hyperchromic anemia, then really the first thing we do is iron studies because we need to differentiate it from iron deficiency. If it's not iron deficiency, then we go along and do hemoglobin electrophoresis, and that is able to differentiate hemoglobin A2, hemoglobin F, and hemoglobin A. And so you can see here, depending on the results that you get, it can be an indication of the sort of thalassemia that you have, beta thalassemia, alpha thalassemia, hemoglobin H disease, or other hemoglobin variants. And then, of course, you go on and do genetic testing or DNA testing, molecular testing. On this slide, we've got a comparison of results between iron deficiency anemia and thalassemia. So this is thalassemia trait, alpha or beta. And so the person has clinical signs and symptoms, but they're not severe. So they both have a reduced MCV and MCH and the NCHC. Serum iron concentration. So these are the iron studies here. You can see that in thalassemia, they're normal or slightly increased in the case of serum iron, whereas with iron deficiency anemia, they are quite different from normal. And then when we come down to the electrophoresis, we can see that in iron deficiency anemia, the electrophoresis is going to be normal. And whereas in thalassemia, we are going to have abnormalities. And again, just pointing out this red cell count. In iron deficiency anemia, it's proportional to the degree of anemia, but in thalassemia trait, it's not, particularly in alpha thalassemia. So here's our hemoglobin H, demonstration of hemoglobin H. And of course, because we do have a hemolytic component to both thalassemia and hemoglobin variants, then we will have other indicators of hemolysis showing us that. Although you can see here that the haptoglobin is usually normal, so it's not sort of enough intravascular hemolysis that it's going to be affecting the haptoglobin. Thank you very much for that. Bilirubin and LDH increasing, so that's telling us that there is some hemolysis there, but it's usually extravascular. And again, you've got fragile cells moving through the spleen. So the treatments and therapies, so in alpha thalassemia here, this is our alpha here, and that's an alpha there as well. So if we have 1 or 2 gene deletion, then really treatments or therapies are not required. There is enough alpha chain production for the person to live quite normally and quite happily. It's really only when you get to the 3 gene deletion, so where we have our hemoglobin H, that things may go awry. So we can, as I said before, you can have transfusion independent and transfusion dependent forms, or it could be that the person just needs to make sure that they've got enough vitamin supplements because we've got increased turnover of red cells, so we need increased nutrients to supply the bone marrow. When you're looking at beta thalassemia major there, you can see that there are a number of things that we can do. We've talked about the reason for splenectomy before, and so that's really to try and prolong the life of the cells that have made it out of the bone marrow. And the gene therapy down the bottom there, that's really quite exciting and innovative. And I don't know how many of you watched the presentation about molecular and cytogenetic analysis of abnormal hemoglobins that Steve Myers presented, but it talked about current research into turning on locus control genes. So those of you who are doing molecular biochemistry will be learning about those. And so these are genes that allow access to chromosomal regions for transcription and translation. And it could be that these genes have been deleted or silenced. And so opening up those genes again is one way of being able to correct the molecular defect. And the other thing is actually work's being done to reverse the gamma beta switch. And so if we can reverse that switch and so get more gamma chains being produced, then we can at least have the person producing fetal hemoglobin to help with their condition. There's a number of other thalassemias that you do need to know about, and the reason that you need to know about them is because they're a little bit different to the normal run-of-the-mill alpha and beta thalassemias. So the alpha thalassemia you need to know about is hemoglobin constant springs, and this is unusual because it actually comes about due to a point mutation. So you may recall that most alpha thalassemias come about due to a deletion, gene deletion, but this one is a point mutation. And so what happens here is that we get an elongation of the alpha chain, and it results in, it's due to additional bases being added to the end of the messenger RNA. So the elongated RNA is very unstable, so it only produces a small amount of hemoglobin constant springs. And so it's very difficult to actually detect this abnormal hemoglobin, but you can do it by alkaline hemoglobin electrophoresis. I know, the reason you need to know about hemoglobin constant springs too is that it's quite a common mutation in Thailand. So that's another reason that we need to know about it. So the beta thalassemias that you need to know of, there's one called hereditary persistence of fetal hemoglobin, and due to a deletion in the delta beta region that you can see there, we get increased production of hemoglobin F. Let me see on my notes here. So hereditary persistence of fetal hemoglobin and delta beta hemoglobin, so the second one down here, they're very closely related. They're both due to deletions in the delta beta region, and so that affects the production of hemoglobin A and hemoglobin A2. So concentrations of hemoglobin F stay high after birth and into adulthood. And so that's why we get this increase in persistence of hemoglobin F. Hemoglobin Lepore, this one down here, is a rare variant of delta beta thalassemia that you see there. And the unusual thing here is it's a result of a fusion of the delta beta genes, whereas the other two are a result of a deletion in the delta beta region. So that's a little bit different there. And so you get the heterozygote and the homozygote type that you see as a result of the fusion of these genes mimics very much the beta thalassemia minor phenotype and the beta thalassemia major phenotype, so they need to be differentiated. Okay, there's a summary and some review questions for you. So I thought we might take a 5-minute break here so that you can get up and stretch, go and make yourself a drink and give my voice a rest for 5 minutes. Has anybody got any questions? Either through typing or through unmuting your microphone and saying hello. Okay, so what we might do is we will take a break, and I will stop this recording, and then we'll come back in 5 minutes time. So it's 4:02 now, or 4:03. So if we come back, say we come back at 4:10.